Hello, and welcome to Nature Connect's TNC in Massachusetts webinar series that connects you with scientists, experts, influencers, and our partners from New England and around the world. Thank you for joining us today. I'm your host, Tricia K, Offshore Wind Policy Manager for the Nature Conservancy. Before we get started, I want to acknowledge that the result of our recent presidential election is raising questions for people about the continued importance of offshore wind under the incoming administration. From the time of our very first webinar in July, we have been talking about the significant role that offshore wind has in meeting our energy needs and providing energy security. Electric utilities have doubled their forecasts of how much additional power we will need by 2028. This is especially true along the Atlantic coast. Until there is another realistic means of meeting this demand, the Nature Conservancy is not wavering in its conviction that offshore wind will continue to be a must-have energy resource and that we must continue to support the responsible siting and construction of offshore wind projects. Part of responsible siting and construction means that we have to continue to learn and improve on our understanding of the ocean environment. In fact, in recent years, we've seen an explosion of new research and monitoring efforts in the ocean along the Atlantic coast and have access to more data as a result of offshore wind development. Today, we will be talking specifically about how we identify research needs and build our knowledge base about the effects of offshore wind construction and infrastructure on fish and fish habitat. In the weeks leading up to this webinar, I've asked several fisheries and marine scientists why it's so hard to study fish and get answers about the effects of offshore wind on fish. And interestingly, they each answered my question first by quoting an oft-quoted fisheries science, John Shepard, who said, "Counting fish is like counting trees, except they're invisible, they move around, and they eat each other." In other words, the ocean is a deep, dark, and unhospitable environment for research to begin with. But then, if on top of that, your research subject is fish, influenced by warming ocean temperatures and a slew of other ocean pressures, determining cause and effect is not an easy undertaking. Despite these obstacles to learning, we still have to try to understand the interactions between offshore wind and fish for some very important reasons. First, our commercial and recreational fisheries in the United States are considered a public trust resource. Sustainable fisheries have made up the cultural fabric of our coastal communities for millennia. Fisheries contribute to our food supply and our local economies, and the health of our fisheries supports the basic functions of a thriving ocean ecosystem. And like birds, fish are also important indicators of consequential changes caused by climate change, pollution, and other stressors that we need to be paying attention to. Even though the starting point for our understanding of fish is dramatically lower than our understanding of species on land, off the Atlantic coast from Maine to North Carolina, we have perhaps the best fisheries data of any other place in the world. This means that we have great opportunities to design our ongoing research to specifically look at the interactions between offshore wind and fish. And it also means that we have scientists along the Atlantic coast who are experts in asking the right questions and carefully designing research studies to get answers we can rely on. We are incredibly fortunate to have two of these experts with us today. We're going to talk about some of the questions they're asking and how they're designing their research to study cause and effect for fish and fish habitat. First, we're going to hear from Dr. Annie Murphy. Annie is a biogeochemist and principal scientist at Inspire Environmental. Her expertise is in coastal and marine systems. Annie leads benthic assessment teams and designs benthic monitoring programs for offshore wind. When we use the word benthic, we're talking about the flora and fauna found on the ocean floor or in the bottom sediments. She'll be talking about how benthic habitats support fish and how offshore wind companies designing monitoring plans to look at the effects of infrastructure on benthic habitat and function. Dr. Brendan Runde is a marine scientist at TNC's Virginia chapter. His work focuses on strategically improving fisheries data collection, understanding interactions between offshore wind and fish, and using this information to help ensure sustainable populations. He's going to talk about a first of its kind study that he is conducting in partnership with the National Oceanic and Atmospheric Administration, or NOAA, to investigate fish behavior in response to offshore wind turbine installation at the coastal Virginia offshore wind project, located 27 miles off the coast of Virginia. As you think of questions for our speakers, please enter them in the Q&A box at the bottom of your screen. We will hear from both our speakers first before the Q&A portion of today's program. Thank you all so much for joining us today. Annie, I'll pass it on to you to get started.
Okay, great. Thank you. Let me share my screen. Okay, thank you, Tricia, and thank you, TNC, for hosting this webinar. I'm really happy to be here. As Tricia mentioned, I'm Annie Murphy. I am a principal scientist at Inspire Environmental. We're based in Newport, Rhode Island. We're part of a larger company called Venterra Group, which is based in London. And I'm really happy to be here to talk a little bit about the interactions between seafloor habitats and offshore wind development. So I consider myself an ecosystem ecologist, and as an ecologist, I think a lot about, and I do a lot of work thinking about understanding how the benthic environment functions. And as Tricia mentioned, the benthos, it's actually a Greek word meaning depth of the sea, and the benthos encompasses the seafloor and all the organisms and species that live in and on and in close association with the seafloor. And the benthic environment serves actually really important functions in the marine environment as a whole. It serves as important habitat. This means it provides food and refuge resources to other organisms and many species. It also serves important functions in terms of nutrient and carbon cycling. The image on the left is a benthic habitat that's composed largely of coarser sediment, small cobbles and rocks, that's encrusted by epifauna, the organisms that live on the structure, those rocks. The image on the right is a sediment profile image, and this is showing a muddier, sandier environment, softer sediment. What you can see from this is a really dense carpet of tubes that are growing at the sediment water interface. And these are amphipods that are serving a really important function, shuttling organic matter, particulates from the water column to the benthos and helping with cycling nutrients in the system. In general, ecologists think about complexity, and the complexity of the sea floor generally is thought to increase habitat value. And by complexity, I'm talking about the variety of different sediment sizes, rock sizes, boulders and cobbles and pebbles mixed in with sand. This variety of structure provides diversity of spaces, different niches that organisms can exploit or use as habitat, hiding spaces, and a diversity of different food resources that can be used. Now here on the US East Coast, the broad continental shelf off the coast of the United States on the east side, eastern seaboard, is the Northwest Atlantic continental shelf. It's mostly characterized by soft sediment. So the image here, it's a sandy habitat, low complexity, but it's also pretty valuable to some species like this squid and sand dollars and other species that are typically found as in fauna, those organisms that live in the sediments like clams. We know that really for generations, human activity has been affecting and altering benthic functions in different ways. We're here to learn about offshore wind, and really one of the most profound effects of offshore wind development in the marine environment is on benthic function. It's this habitat conversion, converting that sandy bottom to rocky bottom and introducing a structure that literally spans from the surface of the ocean to the seafloor. So on the left, you see a monopile that was installed about 2 months before the image was collected, and it's covered in hydroids, these animals that are encrusting that new surface. On the right, you see the seafloor at the base of a monopile, an offshore wind turbine, a different one than the one on the left. And again, that material is being colonized and encrusted by epifauna, high coverage and density and diversity of epifauna, those species living on the rocks. And if you look carefully, you can see some other organisms, some fish. So before we get to those monopiles that steal in the water, there's a lot of work that's done before that happens. So here's a really overly simplified, if you can believe it, timeline of a typical US offshore wind project. It starts with the federal government and states really trying to figure out where to site these wind farms. This takes many years and input from a variety of different groups and people and experts. And at this early stage, the seafloor characteristics and the habitat there is considered at this very early stage. As the areas go from those broad call areas, whittled down to wind energy areas, and then finally to delineated lease areas. Once those leases are identified and auctioned off, offshore wind developers collect even more data on the seafloor, using high-resolution multi-beam and side-scan technology to map the seafloor of the leases that they now are leasing. And they couple that geophysical data with other types of data like images of the seafloor and physical samples of the seafloor to create these detailed maps of the benthic habitat present in their leases. And this information is used to infer about what types of species are using those habitats. And it's really the basis by which environmental impact assessments are done. This is the baseline. These environmental impact assessments are really formulating the hypotheses of what we expect to happen to that baseline, to that benthic habitat with the proposed activity, with that development of offshore wind under different scenarios. And then once that company receives a permit to build, after all of that permitting, those permitting steps, there's more data that are collected, looking at those expected changes and whether there are any unexpected changes that we see, adverse impacts, for example. Okay, so don't worry, I'm not going to get into the nitty-gritty of all of these research papers. I put these up really to demonstrate that there is a lot of research happening in this space, and specifically across the globe, understanding how introducing offshore wind structures influences benthic habitat functions. This is actually a really small smattering of research papers, and these were all published within the past year or two. And there's many more other studies. So the idea, or what I'm trying to convey, is that we have an understanding, we have an idea of how this activity changes benthic ecosystems and the organisms that rely on these benthic habitats. And to summarize, what I really think about is when we start introducing novel or new structures to the marine environment, this does lead to ecological shifts. We've seen this on other spots and other contexts. And these changes can be thought of in two general buckets. One, and they're very much related, almost the same, basically. So one is shifts in species distributions, and the other is shifts in energy or organic matter or carbon or food distributions. So in terms of species distributions, we introduce structures that changes who lives in that space. You now have a new structure that again extends from the seafloor all the way up to through the water column. That's a new space that benthic species can colonize. There are studies and hypotheses that have suggested that these structures may serve as stepping stones for species that are we know that their populations are shifting poleward in response to warming climate. So these new structures may facilitate that process. There's also the potential and the hypothesis that these structures may promote non-indigenous or non-native and perhaps invasive species colonization. So once we shift who's living there, we also shift what those organisms are doing and how they're functioning. So this changes how the energy in the ecosystem moves. So there's a redistribution of energy or carbon in this new ecosystem. So most of the dominant species that grow on these new structures are suspension feeders. So they're epifauna that are filtering water and packaging up the phytoplankton that they're feeding on and the other particulates into growing more biomass. That biomass can be transferred up the food web into higher trophic levels. Those organisms are also facilitating and packaging that material into waste that gets shuttled to the seafloor. Here is some imagery of one of the research turbines down off the coast of Virginia. This was collected about 2 years after the structure was installed. You see it's coated in a dense population of blue mussels, some red algae. These mussels are those suspension feeders. You can see some of them are gaping, they're open, they're filtering the particulates from the water column and feeding on that and growing. Again, some of that material is transferred to the fish using that habitat, and some of it is shuttled to the seafloor where it's used by other species and microbial communities. Here's a deeper portion of that same monopile. So the previous one was about 12 meters. Now we're at about 20 meters or about 60 feet. So we're turning around and we're looking at the base of the monopile. So there's structure that's installed. These boulders are installed. These cobble are installed at the base of the monopiles to prevent scour. So this is a new reef that's been installed at a previously sandy environment. And these fish are utilizing that habitat. And Brendan will get into that in a little more detail. So we have these hypotheses. We understand how what changes are expected, but of course this is a new venue. We've never installed offshore wind in this area. So it's important that we do collect monitoring data post-construction and to really understand it fully. And to do that, we're developing and implementing really carefully designed monitoring programs designed to detect changes in the benthic function. Here I'm just showing a few tools that are typically used depending on the hypothesis or the question being asked and being monitored. All of these tools are non-extractive. So this means that we're not disrupting the species. We're not catching the animals, killing them and counting them. We're relying more heavily on imagery like the sediment profile imaging system in the top left. And video, so collected either through an ROV, towed video sled, or small underwater drones are super useful. And also baited remote underwater video. So this is called a BRUV system in the bottom center. It's used to assess really the habitat utilization by fish species and other mobile invertebrates. Finally, I've included acoustic telemetry, another non-extractive tool used to understand fish movement, and Brendan will go into more detail on that technique. So most of the shifts that I've been describing, that artificial reef effect, it's really what I'm talking about are local changes, relatively local scale, a turbine level change. Perhaps a wind farm level change, but in reality, only a relatively small that's only a relatively small space within the context of the greater outer continental shelf here in this ecosystem. So as we continue developing offshore wind and we continue developing monitoring programs to monitor these shifts, it's really important to collect data that can be used to infer about regional scale shifts in benthic function, habitat, and fish use of that habitat. So this is really an evolving process, and we're really, yeah, using monitoring programs, beginning to ensure that these monitoring programs that we're proposing and doing and implementing can be used to fuel things like ecosystem models, another non-extractive tool to infer about larger scale changes. I also wanted to mention the idea of nature inclusive design. So I've been talking about artificial reef effects. There are a lot of conversations going on in the industry and in regulatory resource agencies, conversations about deliberately designing these new introduced structures with nature in mind, and questions about how we may be able to optimize the design of these structures to promote or enhance certain target species or ecosystem functions, benthic functions. This is an ongoing conversation. There are research projects looking at this idea, but at for now, at least we are collecting the baseline data upon which we can assess whether adding things to these structures or designing them in a different way may create more valuable habitat. So in conclusion, there are shifts in how the benthic environment functions following the development of offshore wind farms. This new habitat that's formed really promotes this richly diverse benthic community that's used by a diversity of species. This habitat conversion in some instances can be viewed as positive. Sometimes it can be viewed as negative. It's really a matter of perspective. And for example, if you're a demersal structure associated fish, like this Atlantic cod, this new habitat may be beneficial. On the other hand, if you're a clam, it might not be as beneficial at that local scale. So in spatial and temporal scales matter. So we need to remind ourselves that renewable energy is desperately needed as a way to mitigate climate change. And offshore wind development is one way to deliver that renewable energy. And there are benefits, some benefits associated with these changes and these shifts. And so, yep, that is all. And I'm happy to pass it to Brendan and Tricia. Thanks.
Thanks so much, Annie. That was great. Brendan is going to do a deeper dive for us now into some of the research he's leading and working on, more specifically related to fish.
Okay, great. Thank you, Tricia, and thank you, Annie, for that introduction to offshore wind and the habitat that it creates. I'm going to talk about a specific study that I have in the water underway at that coastal Virginia offshore wind project, 27 miles east of Virginia Beach. I'll begin with a couple of super brief acknowledgements. Once again, this is a study that's funded by NOAA Fisheries, and we're grateful, of course, for the support and their partnership. I've had quite a bit of help in the field, 10 or so people listed on the slide, couldn't have done it without them. And then I want to emphasize that this was a cooperative effort on the water. I'm really grateful to two captains in their vessels, Captain Bill on the Plan Hooky and Captain Bob on the Catherine C. We could not have gotten out there or found our target animals without their help. So to orient everyone, I suspect that most of the people listening to the webinar today are within this area of the United States, but if you're not, a special welcome to you. And the coastal Virginia offshore wind area is shown on the right-hand side of the screen. So that's CVOW. You might hear me say CVOW. That's what I'm talking about, this offshore wind project. And as Annie mentioned, two pilot or research turbines were put in the water right here at CVOW about 4 years ago now. And they are just on the inshore edge of the new commercial scale project that Dominion Energy is building. So we call that CVOW or CVOW C. And when finished, that commercial scale project will be just short of 200 turbines. The study that I am undertaking at the CVOW area is in an attempt to evaluate the behavioral responses of fish and invertebrates to construction noise. What you're looking at here is a figure from a paper that's a couple of years old now that shows just a few of the potential sources of sound throughout the offshore wind development process that could be heard or otherwise could impact animals underneath the waves. So what I'm going to focus on is the middle section here, which is the construction phase, the actual installation of the foundations of the turbines. And that, for most offshore wind projects, including CVOW, involves what we call pile driving. So using vibratory and impact devices to slide this foundation, this turbine base into the sediment, into the seafloor. Here's an aerial image from Dominion Energy that was taken this summer during the construction of one of the commercial scale turbine foundations at CVOW. So this green vessel here is the pile driving vessel, and you can see that they have one foundation sort of gripped in the arms of the vessel, and there are a couple of more foundation components right here that are waiting to be installed. I'll also point out the 2 rings of bubbles on the surface of the ocean here. That is a noise mitigation technique called a bubble curtain. So the developers are taking action to attempt to mitigate the noise that's created from this turbine installation such that it has a lower effect on the marine life in the surrounding waters. Okay, so on to the fun stuff, on to the fish tagging. What you're looking at here is a sketch of a black sea bass that is showing the tag attachment method that I used for this study. What do I mean by tag attachment method? Well, we used electronic tagging, otherwise known as telemetry or acoustic tagging, to monitor the movements of fish after they were tagged and released. In this image, the green spaghetti tag or streamer tag is shown as inserted through the dorsal musculature of the animal and passed through the end cap of the acoustic transmitter. So this acoustic transmitter is maybe the size and shape of a AA battery or like a chapstick tube, but a little bit shorter than a chapstick tube. So we use this external attachment method because it's fast, it's less invasive. It allows us to get the fish back in the water faster so that we're less disruptive to their regular daily activities than would be a tagging method like using surgical implantation. So for this study, we went fishing at the pilot or research turbines at the CVOW area. So in both of these images, you can see a turbine. Here, it's far off, but we were actually at the base of the other one when we caught this black sea bass and tagged it. And here you can see, obviously, we're fishing right next to the base of this one turbine at CVOW. And as Annie mentioned, the base of these turbines consists of that rock or rubble material that's used to prevent scour or sediment movement. And that does provide really good habitat. And so we fished right there because we knew, based on Annie's work and the work of others, that the animals we were attempting to tag really like to hang out on that structured habitat. So we caught these fish, we put these tags on them, and then we released them right where we found them, right at the base of these turbines. We also tagged 15 channeled whelk. Channeled whelk are a commercially important invertebrate species that lives on the seafloor in the area. And so throughout the tag season of 2024, which was about April to about August, we tagged 72 finfish, and most of those were black sea bass, and then 15 invertebrates, 15 channeled whelk. And again, we picked these animals because, at least for the finfish, we expected that they would stick around. We needed these animals to stay at least in the general vicinity of where we tagged them on this rubble field, this scour protection, because that's where we had our listening devices, and we needed those tags because they're acoustic tags. We needed those transmissions to be close enough to be picked up by the listening devices that I'll show you in the next slide. So these listening devices, otherwise called receivers, are the black cylindrical devices that you see in both of these photos. They're anchored to the seafloor in the area of the two turbines. You can think of these receivers as cell phone towers. The receivers are the cell phone towers, and they are listening for the cell phones. And the cell phones are the tags on the fish. And just like your cell phone, you have to be close enough to a cell tower in order for the transmission to occur. So when we have a tagged fish in the area, it needs to be about maybe 300 or 400 meters from these receivers in order to be picked up. If a transmission is picked up by 3 or more receivers, we can then triangulate the position of where that animal was when the transmission or ping occurred. A little bit of a busy figure here, but I just want everyone to focus on the top, maybe the top third. So what we're looking at is the grid of receivers that I placed around the two turbines at the CVAL pilot area. The white icon with a turbine in the middle of it, that of course are the two turbines in the CVAL pilot area. And the triangular objects on the screen are representing the receivers. So you can see that I had sort of a cluster of receivers around each turbine with a few in between. And these were very carefully positioned such that we had a good chance of picking up each transmission or each ping on 3 or more receivers if the fish was in the area. You'll notice some other colored and shaped icons on the screen. Those are just different devices that we had in the water to pick up ancillary information like temperature and noise level and things like that that helped us out interpreting the results of the study. Okay, I'm going to jump into a little bit of actual hard data here. So the type of things that I'm looking at when I'm attempting to infer the behavioral changes of these tagged animals when other things were happening around them, like offshore wind construction. This is a busy figure, but I'll walk you through it. Each black dot on the screen represents a single transmission from a single animal. So this is just one fish that we tagged. This is, as you'll see at the top of the screen, this is black sea bass number 5108. 5108 is just the tag number. Each ping tells us where that fish was at the time that the transmission occurred. On the x-axis or horizontal axis, we have time from mid-April when the fish was tagged, that's what the green arrow is showing, to early October when I pulled the receivers out of the water and ended the major part of this study. Critically, on the y-axis, the left vertical axis, we have depth. At the top of the screen, represented by the blue line, we have the surface of the ocean. At the bottom is the seafloor. That's represented by a brown line. And in this area, the seafloor is at about 26, 27 meters. These tags, they give us the depth of the animal with every transmission. So right here, we're just looking at one-dimensional, well, two, the second being time, but just the depth movements of this animal from mid-April to early October. So what we can see here is that overall, the bulk of the pings, the bulk of the detections for this animal are quite close to the seafloor. They are at maybe 23 meters and below. So that's representing when the animal is probably on the scour protection around the base of the turbine, maybe coming up a little bit. In one of Annie's videos, that you might have noticed there was a cage-like structure around the base of the turbine. That's an anode cage that creates a lot of good habitat for fish like this black sea bass. And then you can see that there are periods of time where the animal comes quite a bit off the seafloor. For example, in mid to late May, we see one instance at least where the animal comes up to about 5 meters below the surface. This is pretty far up there, and we know from videos like the one that Annie showed that these animals, they're hugging the turbine, they're hugging the tower, and they're using that extent of vertical habitat for foraging and seeking refuge from larger predators and things like that. So we can see that there's this period in April to May where there are a lot of these vertical forays up the side of the turbine. And then from mid-May to really like mid-September, we only see a couple of those. This is when pile driving was occurring during the summer. Now, I'm not suggesting that this behavior necessarily changed because of pile driving. I'm just saying that pile driving was occurring. This could have been just a normal seasonal change from this one black sea bass. And we do have other black sea bass where we have data of them coming up in the water column during the summer. So we're not drawing any conclusions from this. I'm just showing you what the type of data are that I'm looking at to infer how the behavior of these animals was or was not changing. All right, now I'm going to show you a different type of data, and I'm going to show you 3 different one-day snapshots. They're all going to be from the same fish, black sea bass 39. The one I'm showing you right now is from May 17th. That's 5/17. And on the x-axis, you can see that it starts at midnight on the 17th and it goes to midnight on the 18th. This is exactly a 24-hour snapshot. There are 2 data streams that I'm showing you on the slide. One of them is taken from the tag directly, and that is, once again, the depth of the animal. That's the black line. And so what I'm going to tell you now is that in the morning of May 17th, you can see that this particular black sea bass 39, was hanging out at the seafloor. It was right there at 26, 27 meters. And it really wasn't coming off the water, up into the water very much at all. And then in the morning when the sun came up, this is UTC time, so 4 hours later than Eastern time. Nevertheless, when the sun comes up, this particular animal comes pretty far up in the water column to 15 to 10 meters under the surface of the ocean. And while it's daylight, it's moving around. Maybe it's foraging, maybe it's avoiding predators, but it's moving around quite a bit up and down in the water column. And then when the sun sets later in the evening, it does return to closer to the seafloor. Now, the red data stream that I'm showing you is an estimate of speed of this animal. Now, this is derived from the X and Y movements, the position of the animal that is triangulated from the receivers in the area. So it's a little hard to see, but that red line in the morning when the animal is at the seafloor, we're not seeing a lot of blips in that speed. We're not seeing much above maybe like 0.1 meters per second, and that's our units of speed here. However, as we see when that animal comes up in the water column, we are seeing quite a few more speed blips that are higher up. We're seeing maybe fewer blips that are right around 0. So obviously this animal is moving around during that period when it's up in the water column. Now this day, May 17th, there was no pile driving occurring. This is before the commercial CVOW project started construction. Now here's the same fish on the 26th of May, so 9 days later. This looks a lot like the other slide that I just showed you, except we have 2 new colors on this slide. We have vibratory piling, which is represented by a gray bar, and we have impact piling, which is represented by an orange bar. These are the 2 main processes by which the developer installs the monopile foundation into the substrate, into the seafloor. Now, I don't really see anything here. This is just qualitative, but I don't really see anything here that suggests anything different than what I showed you in the prior slide. Again, this is the same fish, it's just 9 days later. It spent most of its time near the seafloor in the morning, and then during the day, it came up and down in the water column. The speed or velocity red line tells us about the same thing as on May 17th. And then in the evening, maybe a little different time of day, but a couple hours earlier, it comes back to the seafloor and settles in for the night. What you can't see in this figure is what I'll tell you now, which is that, there we are. The pile driving distance from where this animal was on May 26th was 2.7 miles. So that's fairly close, but it's not right next door. Now I'm going to show you the last we heard of this fish, which is on May 31st. This is another pile driving day. And something different happened here. And so the colors should be familiar. In the morning, the animal was at the seafloor around the base of the piling, doing its fishy thing. For some reason, we don't see, for this fish on May 31st, we don't see it come up into the water column. I'm not quite sure why, but then what we see in the afternoon or early evening when pile driving is ramping up, we see this fish disappear. We lose track of this fish, and it actually never came back. And it left the area. So why did it leave the area? Well, I don't know, but what I can tell you is that pile driving did occur on this day, on May 31st, and it was actually occurring at just 1 mile from the turbine, so 2.5 times closer than the previous one. That means that the noise levels were probably a little bit louder. Maybe the vibrations in the substrate were a little bit more. But this is an early analysis. This is just a qualitative look. Did the pile driving on May 31st cause this animal to leave the area? I don't know. We're working on answering questions like that. But I want to stress that this is correlative. It's not necessarily causative. We had other tagged animals at the same turbine on May 31st that did not leave when pile driving happened. So part of the analysis that we're working on is trying to piece apart how these animals behaved and why they behaved, and whether that any change in behavior like emigration that you're seeing here was in response to the construction activity. So stay tuned. So next steps, data analysis is underway. This study ran from February to October. We're hoping to have some shareable results by the spring of this coming year, 2025. And meanwhile, we continue to monitor for tagged fish at CVAL. I left a few receivers in the area, so if fish like that one I just showed you come back, the tags have a battery life that should last maybe 6 to 8 more months. If it comes back, we'll hear it, and we'll be able to say that that fish came back. So we still are giving ourselves the opportunity to collect more information. So I want to end on this, and this is the so what. Why are we doing this? The response that I just showed you, if it was a response to pile driving, that fish left the area. That's a sublethal response. Clearly, that fish did not die. If it had died, we would have seen very different acoustic signature there. So why should we care about the effects of offshore wind on fish, even if those effects are sublethal? Well, fish populations, they're a public resource. Like Trish mentioned, just like other wildlife species, waterfowl, white-tailed deer, even non-game species, like one of my favorites, the red-headed woodpecker, it's all of our responsibilities as citizens and my responsibility as a scientist and a conservationist to work towards a better understanding of how human activities influence individual and population level responses of these animals, because they are resources. Better science leads to better construction practices, and better science leads to more appropriate mitigation where mitigation is necessary. So I'll end on that. There's a QR code on the screen if you're quick. That'll take us to the Nature Conservancy Virginia website. I'm going to have a link shared in the chat that will give you more information about the study that I just presented on. And so now I'm going to stop sharing, and I think hopefully we have some time for a question and answer period.
Thanks so much, Brendan. And yes, I hope we do, because we have a lot of, as usual, a very engaged listening audience and a lot of questions to get to. So before I go to the Q&A, though, I just wanted to ask, I guess, a couple of quick questions following on some of the things that both of you said. So first to Annie, I'm curious about, you talked a lot about like the monitoring plans and the monitoring effort, and I guess I'm curious about learning more about who's responsible for the monitoring after the wind project is constructed, like who funds that and who supports it, and whose obligation is it to produce and share data from that ongoing monitoring?
Yeah, great question. And it actually, that monitoring has to start before construction, right? So you have to get that baseline in the design that the monitoring plan or monitoring program is designed. And so offshore wind developers are required to develop a benthic and fisheries monitoring plan for their lease area. And their boundary is their lease area, their area of impact. And so that's their scale at which they're monitoring change. And those monitoring plans get approved by resource agencies and vetted and comments and are developed very collaboratively. Those monitoring plans there are other avenues in which research is being conducted. So some power purchase agreements require offshore wind developers put resources into a funding pot of money somewhere, depending on the state. Those states are then distributing that funding to researchers to answer more regional questions and targeting different types of monitoring, or using that developer funded monitoring program at the lease scale to answer more regional questions.
Great, thank you. And Brendan, what happens if one of your tagged fish gets fished, gets caught? You follow it home and invite yourself to dinner? Like, what goes on with that?
Yeah, great question. So the tags, the streamer tags, which in our case are red, there was one figure I showed where they were shown as green. They do have a phone number on it. It happens to be my personal cell phone. So my friends and family chuckle because when they call me, it says, "You've reached Brendan Runde with the Nature Conservancy. If you've caught a tagged fish, blah, blah, blah." And what we'd like people to do is do whatever is legal. So if the fish is sublegal or out of season, that fish needs to be released. If it is a legal size fish that is within season, you're welcome to retain it, take it home, have it for dinner. I have received quite a few, maybe 5 or 6 now, tag returns from folks who have caught our fish and given me a call. It's always great to have that information. We know what happened in those cases when the fish leaves the array.
And can you follow up on sea bass in particular? Do they have a swim bladder that's affected when they're brought up to the surface?
It's a great question, and I appreciate somebody asking that in the Q&A. They do. So we know that black sea bass and other fish that have a swim bladder, they experience what we call barotrauma or pressure-related injury when they are brought to the surface. And this can cause an expansion of the gases in the body cavity, in the swim bladder and other organs that can at times render a fish unable to resubmerge. In other words, they would be trapped floating at the surface. So when we released our tagged animals, we used what's called a descender device. That's a barotrauma mitigation tool that takes the fish back to a depth where those gases recompress and the fish can swim away. I'm going to put a couple links in the chat if you're curious about barotrauma mitigation, specifically with black sea bass. There's a link, and then I'll put one more in there while I let Trish ask the next question.
Great, thanks. So, and this, I think both of you could probably answer. This is a question by someone in the Q&A, and it goes back to kind of where you were starting to talk about, Annie, but this idea of baseline data. This particular question is specific to fish and certain marine species and in what density. So it's going to like the often raised concern by commercial fishermen that we don't have great baseline data about certain fish. So if we don't, how do we know what's being harmed if we didn't measure it up front really well? So wondering if you could talk a little bit about baseline data and how important it is and the techniques and methods that scientists use to make sure that we have enough baseline data. And I guess I would kind of translate this question to going beyond fish and also just baseline data for you, Annie, about the habitat itself and what constitutes good enough. But maybe you can both kind of take turns, speak to the fish and the habitat.
Sure, I can speak to the habitat. So the baseline habitat data is pretty sparse before the leases are auctioned off. And then the developer, for engineering purposes, but also for ecological impact assessments, collect high resolution seafloor mapping data, that geophysical data, and point collection of imagery. So I think at a benthic habitat level, we have a really good understanding at that lease level scale what exists as baseline and how that habitat's functioning. Maybe Brendan can talk more about fish densities and the baseline of fish.
Yeah, there are some monitoring studies going on at CVA that I'm not involved in as part of the fisheries monitoring plan for that project. What I'll say about my work is that the baseline that we needed for our study was on the individual level. So we started tagging before the monopile construction began. So we have individual baseline data that tells us how an individual animal was behaving before construction began. So that's the type of baseline data that I deal in. But you're absolutely right. It is a big challenge from a fish density or fish abundance standpoint to try to collect enough baseline data.
Do you, can you talk, again, this is for both of you can weigh in, but can you talk a little bit about like the role of the companies in monitoring and collecting this data both before and after? Like, do they benefit from this? Do they invite these kinds of research monitoring studies into their leased spaces? Are they all required to do the same type of monitoring, or do they decide kind of project by project? I'm just curious to know like the role of the companies and their viewpoint on whether this is something that's beneficial and they want to contribute to it, or that they just have to do it. And
To me, it
You
depends
want
on the company. But for the most part, in my experience, these offshore wind developers are very much pro-environment. They are in this industry for a reason, this renewable energy industry. And there are corporate goals that various companies have to document their environmental stewardship, and they see these monitoring plans and these monitoring programs as a way to show that they're developing this resource responsibly and environmentally responsibly. So, and in terms of, are all developers required to do the same monitoring? That no, there are very there are not very clear rules, guidelines, and for good reason. It depends on where you're building. So you want to be able to shape these monitoring plans based on where you're building. It shouldn't be a cookie cutter, you have to do all of these things. And then once we understand one process, we don't need to keep asking that same thing over and over again, as long as we understand it. So I think there's a reason why not every developer and not everywhere needs to collect the same type of data, but there definitely should be some standardization. That being said, because we want to use that data to think about regional scale changes, and the developer is required to collect data within their box, and that's what they do. We're limited in that space that we can do it. I will say that the developers are hiring academics. They're hiring a lot of researchers to develop these plans for them, and consulting agencies. And so they're going to the experts to answer these types of questions. And I'm hopeful that we're doing the right thing.
and Tricia, I know you want to move on. I'll just
go ahead.
add
I'd love to hear from you.
Thanks. I'll just add that NOAA Fisheries is undertaking an effort right now to streamline and align the fisheries monitoring plans among projects, such that there are some cumulative effects that we can start to look at. If the types of projects are standardized in terms of the data they collect, then the results are not restricted to a single project, and that can be very useful from an ecological standpoint.
Have there been, this might be more of an Annie question too, but have there been studies that compare or that are looking specifically at the shift to more complex hard bottom habitat versus you're referring to it as, you're calling it habitat conversion? Like, are we losing important ecosystem functions when we move from soft sediment habitat, sandy bottom habitat, to more complex? And are we designing studies to try to know the answers to that?
Yeah, there's a study that BRI is leading that is more of a food web study, and looking at what are the food resources that sandy bottom is providing, sandy habitat, and then what are the resources that that hard structure is providing. And so the fish that are using that food are quite different, and who's using the soft sediment and who's using the hard sediment. So or the hard bottom. It's definitely an ecosystem modeling question that we can get at with an ecosystem modeling approach. And that's one study that comes to mind that is ongoing.
Great. Brendan, we often get questions like this when we're tagging animals, just about like, are there what are the impacts to the tagged fish, and are there approaches that we use when we're attaching these acoustic tags externally versus internally to, to make sure that we don't have more negative impacts?
Yeah, got it. That of course is an important consideration because we are assuming that the tagged animal is behaving the same way as an untagged animal. We're assuming that the tag has a negligible or zero impact on the animal. And so the tag attachment method that I presented today, I performed a study I live in North Carolina, here in North Carolina a few years ago, where we tested 6 different attachment methods for this type of transmitter, actually on black sea bass. We tested it in a tank. We looked at these different methods. We looked at how they influenced tag retention, which is whether the tag stayed on or fell off, whether the animal grew at the same rate as untagged fish or fish that had different types of tags. And we looked at animal welfare. We performed histopathology. We had a semi-quantitative scoring system of how the tag as an externally attached device influenced the animal. And so I'm going to ask one of our folks to drop the link in the chat that I just shared with them to this study. It's open access. Anybody can read it. And the tag attachment method that we used in this study at CVOW, it emerged as the best. There was no difference in growth or welfare between the untagged animals in our holding study and the animals that were tagged with this attachment method. So this was not a sort of willy-nilly choice for how to do the tagging. This is a very careful choice because we know that those assumptions are important.
And maybe just squeeze in one more question. Are we replacing structured habitat in places where it's previously been destroyed, like where it existed before? So it's like sandy now, but it was once hard bottom. And does that factor into how we study the effect of kind of whether we say conversion versus restoration?
Yeah, that's a great question. And that's really the narrative in the North Sea. So the North Sea has been largely trawled and overfished, and that natural complex habitat has been mowed over. And so they are kind of thinking of it as a restoration, more introduction of habitat that was used to be there. Here, I think it's a little different. It's a lot larger of a space. We haven't trawled over every square meter of it. So I think there's a little bit of both. We are doing a study looking at how natural boulders and the population and the communities living on natural boulders nearby compare to what the communities on the new structures are, and looking to see whether they're similar. So yeah.
That's great. Well, thank you both so much. This has been so informative. And thank you to all of our listeners for joining us today. And special thank you to all who support TNC so generously. We will be sending everyone a follow-up email with a link to this recording. We appreciate your feedback, so please take a moment to respond to the poll on your screen before you hang up. And it should have popped up. I think it did. And if you're enjoying our webinars, please be sure to share them with your friends and colleagues. You can follow us on social media at conserve_MA on Facebook and Instagram to keep up with our work and get updates on upcoming virtual opportunities to connect with us. As always, you can register for future webinars at nature.org/MA webinars. And during our next webinar in the series, which will take place on December 5th, I will be presenting with my colleague Marta Ribera, a spatial ecologist at TNC, and we'll be talking about what net positive impact on biodiversity means and how we're adapting TNC's marine mapping tools to support this idea. So we hope you can join us then. Thank you all again, and have a wonderful afternoon.